the one thing that is on everybody's mind these days is the price of nitrogen. And I was shocked actually, because obviously I'm not there buying the hi Ian. I'm not buying the nitrogen, and it's sort of like seeing the price of 650 to 700, a ton. I was literally, I was shocked. I was shocked that that it actually gone that far, because I only saw the price from September, which was like 350, and I thought it was high because it was like a 50% hike from a year ago. And then right now it's going up, and nobody's saying that it's actually stopped at that price. So I'm really concerned,
I'm looking at it as an interesting trial into the effects of a carbon tax. Because it's effectively doing what a carbon tax would do.
Yep.
Let's see if it changes behavior.
Mm.
Well, we'll see. I mean, it will be better if there was an actual carbon tax and farmers would actually be getting paid for it, for getting rid of that fertilizer. But I don't think we're there yet. I'm hoping that the people who are working on the carbon markets and the carbon credits, have got something to share with us soon. But yeah, it was a bit of a shock for me. So nitrogen price, that's one thing on my mind right now. I'll tell you basically what's on my mind. What's on my mind is nitrogen price. Second one is driving demand for organic produce. When I say organic, I just basically mean free of agrochemicals. And so, however you want to interpret it. And I have my conversations with the Soil Association. And I know that basically, people have different opinions between organic, regenerative, conventional. Everything is mixed together. Farmers really shouldn't care about these things. Just basically, they're out there trying to make a living, right? From my perspective, I'm just basically here to try and create an alternative to the agrochemical system, and I just think people in general think about organic as that alternative. So I'm just basically thinking of the idea of driving demand for organic bread, for instance, for organic barley, for organic wheat, from the top. So I'm not going to spend too much time on it. I'm just going to tell you that this is something that we're trying to work on from here. So not just trying to solve the issue of access to microbes, but trying to also create a bit of demand for that kind of produce. But again, it's not something that you need to think too much about. The thing that I would like to present to you today is that obviously, you're probably well aware that we want to run a lot of trials next year on spring crops, and we've got a great opportunity to learn a lot about how this biology behaves, what is necessary for this biology to grow a crop to its fullest potential. What do we need to measure, all these things, and we've got. There are people that we could work with to make sure that that it's scientifically valid, and it's also giving us enough data to say that it's going to be economically valid for farmers as well. And we want to do it in the open. We want to do it with farmers. We don't want to hide somewhere in the corner of Rothamsted. So the idea is that with these new grants that are coming in right now, which are a bit cutting short on time in terms of when we can start, but with the grants that are coming in now, we could create a collaboration where there's us developing the microbes, there's Sheffield University that has been very generous with their time, hi Janice, in terms of the oversight and a lot of scientific scrutiny. We would need another partner like Innovative Farmers or AHDB for dissemination of all of this knowledge, all of this information that we find. And we also need a consortium of interested farmers. And so the reason I'm telling you all this is because our initial plan was to simply go out to you and say, well, we're not going to be offering you a farm scale solution because that's just silly. It's, you know, it's too risky for you to be taking on something completely new. But we're going to offer you trials, and you have to pay for those trials. And, you know, I'm looking at the costings and at the scale that we're at right now. Obviously, it's still quite expensive per hectare for us to be running those trials with you. So what I'm hoping, hold on, I'm just going to close the door. What I'm hoping to achieve is that if we get the grant, then the grant covers 70% of our cost because we're just a microorganization. And so the only thing that's left for you is just 30% of the cost, and we're going to try and design these trials in a way that I'm just going to mute these guys. I want to basically design these trials to be as cost effective as possible, and also have, you know, enough replicates for everybody to be satisfied scientifically, but basically to make sure that that every farmer who's participating doesn't have to pay through their nose to understand, you know, how to work with soil biology. So this is just an idea at the moment that we're going to push further, but we'd like to basically invite you to this consortium of farmers that would be taking on a trial with us next year on spring crops, and potentially running these trials for 2 or even to 3 years, because we're just looking at these grants basically and trying to see what timeframes do they allow us to work in. And so I'm going to work on all the costings, and I'm going to make all of these costings available to you. At the moment, the costings that are basically made available on the websites, that's basically what's there at the moment. You can have a look at it, and this is something that, you know, I've worked on 3 weeks ago or 4 weeks ago, but I will redo all of these things so we have a much better, much clearer idea of what the cost should be for proper replicated trials. And we'd like to build a consortium because that would allow us to go to the grant providers and say, look, we have all of these farmers who are interested, and we're going to have a much higher chance of actually getting these grants and actually doing this work. So you would be at the forefront basically of developing, you know, the soil microbial total solution to growing crops or pastures in the case of Judith's. So yeah, that's basically the idea on that. Very quickly, we're always open to receiving your samples, and we're looking forward to putting more people on the composting leaderboard. We'd like to you know, to have as great of a number of data points to have a much better idea of what's happening in terms of, you know, the sort of production of these microbes on a small scale by early adopters in the country. So that's it from me, from the top down, and we can have a conversation now. Any questions?
I just wondered, Daniel, on the scale on an individual site, what are you looking for, and what are you looking for in terms of total number of sites or collaborators?
That's a good question. I basically would like to, I was initially looking at a total of sort of 400 hectares, but I need to redo that. But I'm thinking of, you know, up to 50 participants for that consortium would be something still manageable if we do it, if we design the trials well. And I think we're at a stage where we've got the product refined enough that actually it's very easy to use. And so there would be less oversight necessary from us during application. So, you know, meetings like this may be enough to ensure that the material is applied properly. And there may be some people locally across the country who would be able to provide some oversight as well, rather than us being at every place at all times. So up to about 50. And my initial design was sort of like 5 to 10 hectares with a very small control, but I don't really feel that we should be progressing at this stage with that sort of trial design. I really feel that we need those replicates done properly, but I still want it to be weighed with a combine rather than with a sickle and a scale. So I'd say 2 to 3 replicates per farm, with a whole tramline per 3 replicas. There's Janice raising her 3 fingers. Like 3 beers, thank you. So this is, you know, three replicates. The one thing I'll be asking Janice and your friends, basically, can we limit the control areas which are receiving only water? Because the replicates would be your chemical system versus biology versus control. That's one replicate, and that should be it. But I want to limit the losses, basically. If we're taking the whole tram line, you know, I'm trying to limit the losses. So if academia basically at some point say that, yeah, we could do with one control, I'll be very happy. But if we want to do it properly, we want to have the control with, you know, in every replicate. So I don't know, Janice, could you comment on that, please?
Hi, can you hear me?
I can hear you.
Okay. The reason that we have to have replicates is because out there in any field, there is huge variability. That is the nature of soil. Scientifically, if it's going to be acceptable as in a publishable form, you have to have a minimum of 3 replicates. That is to minimize the variability. If you only have 1 replicate, that might not be representative. So we have to have a minimum of 3. Your scale, which you want to use a combine, so you're talking of a strip, yeah, field strip. I mean, that's probably overkill for the scientific analyses and possibly more risky for the farmer. I would probably only look at 10 by 10 meter plots, which are more easily replicated for those 3 different treatments.
So what I'm also hoping to do with these trials is not just basically to satisfy, you know, the science, but also satisfy like the really practical view of, you know, the scale of application. And you know, we can do sort of, you know, small scale trials, but I think when we actually sit on the machine, and do a proper size. That's why I wanted to aim for 5 to 10 hectares, but then I thought, no,
I can see the sense in that, because it also answers more questions that the farmer would have. Yeah. At the same time. Yeah.
This is exactly my aim here, so that the farmers can actually, you know, see what it actually does, because, you know, we can do it in pots and probably, you know, receive, you know, get scientific. But then when I actually go out with the sprayer, you know, and I spray the soil, and then basically, you know, nothing rains after, you know, for 3 days, and the microbes simply don't survive. If I was injecting it in a pot into the root zone, it's completely different, you know. So.
Oh no, exactly. I mean, I think in your term, pot trials, which would be done in the university under controlled conditions, is to produce an optimum. I mean, I got a lot of questions from your previous session, which thank you for recording, and the questions are all relevant. You know, what's the effect of different soil types? What's the effect of different crops? What's the effect of both of those on the biology, and the biology on both of those? These are very fundamental questions which probably need to be answered under control conditions to ground truth the processes. I mean, you talked in terms of a systematic way that the soil works, but you kind of need to ground truth what is actually happening.
Yes.
Then compare that to the field where you would expect it to be very different, but then you can compare the two and say what would be the likely effects in the fields of different soil types with different crops. So doing that simultaneously probably answers even more questions.
Absolutely. I definitely want to have all scales. The farm scale is really important to me from the perspective of the farmer actually seeing, how it actually works, like in that large scale at the same time.
Absolutely. Yeah, I agree. Yeah.
And I think the other thing that we would be answering, and this is important to me in terms of practical application of this, is that I don't think there's enough machines in the country which are geared towards applying this biology to the root zone where it's supposed to be,
Right.
in the first place, for instance.
Yeah.
And I don't have the answer to that question because I don't have visibility of it. But I think this trial, if we have enough people across the country, would answer that. And so we would be able then to have a better perspective of, what is available to actually practically work with biology.
Yeah.
So
that's also a logistical issue.
Very much so.
Exactly, which is why it's really important if we can get farmers to join the consortium, because they are the end user, the stakeholder, and they're the ones that raise the interesting questions that need to be answered. It's really important if we could achieve that.
Yeah.
What equipment do you need to introduce this biology to the root zone?
Well, I'll give you one example. Last week when we were seeding, where we're drilling, we were hoping to drill with live biology with a coating, and we completely failed with that. It's just, for us, it's impossible to coat the seeds with live biology and still go through the drill. And so I understand that Jake Freestone, for instance, has got a drill with a drip feed behind the drill. And that's one example of a machine that, we only have found. Oh, Andy's raising his finger. Andy, have you got one as well?
I thought I did, but when I did my extract, there's still even the very fine grains that you can even get through a 400 microgram filter sock was blocking my liquid system. So I think even if you've got a liquid system on your drill, you need a peristaltic pump which can cope with the solid part.
What pump have you got now?
I've just got a fairly standard.
It's like a centrifugal pump.
No, it's just what would you call it? I don't know what you'd call it, but just an electric pump on it.
I think it would be great to actually have a look at your rig, Andy.
Yeah, but it's.
Do you have a filter on it as well, by the way?
Sorry?
You have a filter on it as well?
Yeah, I took some of the filters off because they were too small and I knew they were going to block it, but it still blocked the lines, and I was quite surprised. But I understand it. A peristaltic pump is what you need to be able to cope with this kind of stuff, and they're about £4,000 or £5,000.
Yeah, I mean, it's something that we need to. This would be something. This is one of those things that we need to basically, learn. And I think, if we have this, this group together, we could work these things out completely, fully.
George Sly from Horizon Drills might be someone to speak to, because I know he was working with an American company that had all sorts of in-furrow systems that they developed over in the States. I think they're called underground ag or something like that. And there was definitely a peristaltic pump version of that. And it was actually, as you were drilling, it was measuring soil moisture and all sorts of things as you went along. But it had one side of the drill coulter was putting the seed in, the other side was putting a liquid in. I don't think it was biological liquid at that point, but I'm sure it could be adapted to that.
I'd be happy. I'd be really interested, Andy, to see your setup, to understand, where it actually clogged.
It clogged because I've got filters in the lines as well, and if I took those filters out, I wouldn't be able to put I'd have to put too high a water rate on. It's practical because it's basically reducing the flow. That is a little disc filter, And I think that's a good thing about a peristaltic. You don't need those filters to put a lower water rate on. You start talking about 200 litres a hectare with a drill with water rates, it just doesn't work practically. You've got to get down to the sort of 50 litres a hectare at least, I'd have said. And that's what a peristaltic does. But mine, because I have to use a restrictor to get it down that low, and that's where it was blocking.
So that thing is called a restrictor basically for airflow.
Well, it's not a restrictor.
I've just put my email in the chat. Could you, if you have some pictures, I would really appreciate if you could send that.
I haven't got them on me, but I can send you some.
That would be great. Thanks.
Mine didn't work. So, but I probably can put bigger restrictors in, but then the water rate would have to go up, and I don't want to be putting 100, 150 litres of water in every hectare. It just logistically makes it difficult. If you're doing 20 hectares in a day, that's suddenly a lot of water.
Yes.
And weight. So
I think, Andy, they're only £5 a pop, the peristaltics.
As far as I know, that's only for the pump. That's not for everything else that goes with it.
Because I know that Tim's rig was quite a bit more than that.
It might be. I thought that's what Tim told me, but I thought the actual pump, if I wanted to swap the pump only, it was going to be about that. But like everything, that might have doubled in price in the last 6 months.
I heard him say 25 grand for the entire install, but I may be totally mishearing that. So it's great to know.
No, I haven't looked into it, but it's something I was going to look into this winter.
There's definitely the way to go.
Yeah.
Liquid applicator on our drill. John is just saying you've got a liquid applicator on our drill. I can send you some picture.
So another thing, Daniel, actually, that's probably pertinent in this instance is I know that Tim Williams, he's been working with the land gardeners kind of Lubke compost at his farm. He actually said that they drilled just compost using the weaving drill that they've got. No, it's not a weaving, it's a more uni drill, like an unconverted more uni drill, just putting it straight in the seed hopper. I think, as far as I'm aware, they have no liquid application, and yeah, he said it worked all right. So
must have been relatively dry as well.
Yeah, I mean, I think like the material that they're producing is very consistent and it's very fine. So yeah, and it's also quite sort of low volume, and it's being applied at low volumes as well. So presumably they're screening as well. I'm not sure what down to what level basically, but yeah, when you see it, it's a very sort of like very uniform, very kind of like sort of fine grade material basically.
We saw it.
Oh, okay.
We're not putting it on the leaderboard.
Okay. Can we see the numbers?
No.
Oh, okay. They're just too low.
If somebody sends me a sample of their compost basically to test, then I will include it in the leaderboard. But we saw the numbers, and I don't want to comment.
Cryptic.
Yeah. Just to say, we were disappointed when we saw it. We were very excited when we got it, but we were very disappointed.
Well, there you go. Still, I guess in this case, it's the consistency of the material that means it flows through the drill.
Oh yeah, absolutely. This is something that that caused the guys to almost lose it last Thursday, because I told them let's go with an unsifted compost during our extraction process, and yeah, they were quite angry with me because of that. So next time we'll be sifting the compost. Have you considered Base UK as a farmer group to contact the trial members? I think, Sari, did you phone Base UK at one point? I think we need to basically maybe have a different kind of conversation. Is anybody here in touch with Base UK? Andy?
Yeah, I'm a member. I used to be a committee member. I think Ian's a member as well. But they've got 150, 200 members now, all over England and Scotland, and they're all farmers that are doing no-till, cover crops, conservation ag. for all the kind of people that be interested in this kind of stuff.
Uh-huh.
I have some contact details for some of them. Nobody's responded to emails yet that I sent out.
There's a secretary called Rebecca.
Rebecca.
I can send you her details.
Yeah, that's who you want to send a mail out. She can mail out all the members from the one email, and you probably get quite a good response from that, I would have thought.
That would be great. I've also contacted Farmers Weekly and tried to put in an ad, and they haven't responded to me. I think I've contacted them 3 times now.
Mm.
I think we're going to have more success, going through farmers than top down. But if in terms of if somebody like you, Andy, or Ian, maybe speaks to base and say, there's this thing that we want to trial. Maybe there's other base members that could be interested. We'll try from our end, and then maybe there's interest with base to if they get the same kind of voice from 2 or 3 sources, maybe they'll be interested to put it out to other members. What do you think?
I think the best way they do it with everyone else is you contact Rebecca and ask her to put out an email to all members and explaining what you're wanting to do.
Okay.
And with your email, I used it we're doing a research project here, and that was through Manchester Met University, and they got 50 or so replies.
Okay.
I think I'll do the same thing with David, with the NNCGG. I get all the abbreviations mixed up, but yeah.
You should probably also potentially get in touch with the Nature Friendly Farming Network. Martin Lyons, they're doing quite a bit of kind of partnership work, for example. And yeah, it's definitely they're sort of geared up for, it seems that that's kind of, Ian, you were on the walk recently, so you probably have a bit more insights into.
Yeah, I'd say base was probably more appropriate for this particular project. Nature Friendly Farming Network have got a lot of livestock farmers. Certainly not exclusively. They've got some very good arable farmers as well, but they've got an awful lot of livestock farmers, which probably, as I say, base is much more cropping orientated and probably more appropriate for this project.
Does this not want to cover livestock as well, though?
I'm just thinking this seems to be an arable crop trial that you're looking to do. That's all. But as I say, I'm certainly not saying there are no people who would be interested in nature-friendly farming, but as a target-rich environment, let's call it that, I would say base would be a better bet.
The reason Judith is asking is because we've been having conversations about including Judith pastures in it as well, and I don't see a reason why not. The thing is, I've been specifically targeting arable crops because that's just my ambition. I want to have organic bread on the shelf for the same price as normal bread at some point. Maybe it's going to take 20 years, I don't know, and then we're going to call it normal bread. But from what we gather, this biology should be easier to establish, when you've got a pasture. And so, if there are pasture farmers out there who are interested, I don't see a reason why not have a trial associated with this, because we're just working with biology.
I would like to see 2 or 3 out of your 50. I would like to see 2 or 3 pasture-based farms included. I'm not saying half, but 2 or 3. I think that would be valuable. Exactly,
and I think Niels have got some people probably who would be interested. Michael, we've got Michael connected. Is that the same Michael from Scotland?
Yes, that's right. Can you let me just turn on my video? Sorry, just got off work. So, hi guys.
So yeah, basically, Michael could be the person, for instance, helping out in Scotland. So we don't have to go out there. And so if you know of any people, for instance, up in Scotland, Judith, who are pasture farmers, that's something that could be quite relevant, because it's you know, it would be very similar climatic conditions, very similar area.
Yeah, I'll see what I can do. Yeah.
Pastures do get agrochemical SERI.
Need them.
Yeah, in a different.
As simple as, because it's going to be better for the animals anyway. And then we might end up getting organic meat as well, normal.
Well, first of all, Judith is going to hopefully save money on that nitrogen application as well. I seriously, you know, I looked at what's his name, Jeremy Clarkson, saying that you know, him saying in public, saying, I'm not making any money, and I know that nobody else is making money, and he said that before the nitrogen hike. So I'm terrified right now, to be honest, when I'm looking at this.
Well,
So,
I mean, we've had great prices for our lamb and our store cattle this year, and we were on the top of the world, and then looking at the fertilizer price, that has more than wiped out that gain in one fell swoop. And I mean, yeah, we're back to less than square one, just, you know, on that basis. That's without thinking about feed or fuel or anything else. That fertilizer hike has completely nuked any gain we made this summer with the prices we've received. So, and everybody else is in the same boat.
Time to get yourself some foliar in.
say again, Niels?
Time to get yourself some foliar in before that goes up in price as well.
Or just don't use extra nitrogen. Use the soil biology to feed you.
Exactly.
Just going back on the pasture thing, I wouldn't assume that people who are in base don't have pasture as well, or animals, because I know a lot of them do. They just happen to be, I mean, base is generally about no-till and reduced tillage, arable cropping, but a lot of those people are bringing livestock back into their farms for a wider sort of regenerative benefit as well.
Excellent, excellent.
I'd be interested to know, actually, if you're applying this biology and you applied it to a grass crop, would it then still potentially benefit an arable crop if you're using temporary grass in rotation with arable crops?
I don't see why not. As long as, from what I understand, the grazing basically is used to knock back the cover crop before establishing the cash crop, right? You're not actually using glyphosate to knock that back.
They might be at the moment, but you know, if we can build a system around it, because there's a lot of farmers now using grass and grass legume based mixtures through countryside stewardship in rotation on arable farms where they weren't before, and that might be a really interesting place to try some of this stuff as well.
Yeah, it is definitely, you know, we're quite happy to work with naked soils to test this biology, you know, at its extreme. But the best situation is when there's at least some cover crops, you know, before establishment of the cash crops. You know, the ideal situation would be a permanent cover, low growing cover, but I don't see anybody who's successfully figured this out yet. I don't see anybody really doing it successfully anywhere. Some people try with clover and veg, but the closest I've seen was basically somebody having a special mower that runs these little sections of mowers between the rows of the cash crops to bring down the constant cover crop. However, the idea is to have as much actual living plant growth on the surface at all times to be able to support this biology throughout the year and over the years, basically, so it can develop. So yeah, cover crops grazed by animals, it's a wonderful addition, in my opinion. I'm not a regenerative agronomist. This is something that I leave to people like Niels and Ben, and we're just supposed to solve the microbes. But the microbes will get supported with plants and with grazing as well. Stephen, you've got a question.
Yeah, well, I was going to just come in there talking about putting microbes onto the grass and then putting another crop in after the grass. And we were vegetable growers up here in Scotland. I'm very much encouraging them, and they have been putting biology in the autumn onto the pasture. 2 or 3 months later, they then plough, which I don't recommend, but they're growing root vegetables, so they have to disturb the soil far more than is ideal. But getting some biology into that soil early is beneficial for them. They then make their beds, and I get them to put more biology at bed making. They are getting superb results with their organic vegetables. They are getting better yields and better quality yields than conventional growers.
What are they actually applying?
Well, they're actually applying it's not as good as you will be producing, Daniel. It's not a decent compost with a good diverse range of microbes. It's simply a consortium of mycorrhiza, other beneficial fungi, and bacteria.
Well, I wouldn't necessarily say that, you know, it's worse than what we produce. You know, if biology, if it's used properly, then it can do the work. And obviously, a lot of people have done a lot of research into these, you know, individual microbes. One of the things that we're considering to trial, you know, during the trials that we want to design, is actually using these bugs in a jug formulations against what we produce, with what we produce, you know, basically all sorts of mixtures. We'd like to learn more about what we're producing, you know, with DNA analysis, to see, you know, are we producing microbes that contain some of these bugs in the jug? Can we basically produce, you know, our material, and you don't have to buy these bugs in the jug? Or do we produce a host of a lot of different stuff, and then they work better if you buy the bugs in the jug? These are the sort of questions that we've got in the back of our mind right now. And, you know, lastly would be, do we buy bugs in a jug, and do we basically add them to our composting process to use compost? As Janice called it, bioincubators, and I stole it, Janice. I keep using it. It's wonderful, I think. And yes, thank you. And basically see if you know, if they can be easily propagated, because they're expensive, obviously, right? So we're looking at all of these options. Doug, you're going to send us some samples from rotational livestock grazing. Thank you. Looking forward to that. Yeah. Okay, got that. Questions, more questions. Who's making compost? Andy's making compost. Andy have sent us some samples. Who else? Niels is making compost. Doug is making compost. Judith?
I need to learn how to do it on a kind of farm scale. I can do it on a garden scale. I need to learn how to do it on a farm scale.
The one thing that we've already discussed, and one of the ideas I've presented to Judith, and this is something that I have said we could explore, is the simplest thing that we can offer at the moment that will be cost effective for farmers who are, you know, too busy to figure out this biology straight away, is to buy bulk green waste compost and buy an inoculum from us. And this could be quite cheap, actually, if it works. So one of the ideas that we have proposed to Judith actually is to buy bulk green waste compost, buy this inoculum from us, and then pay for samples, obviously. We have had some early success with growing out our biology in green waste compost, and it took about 3 weeks for the biology to populate the piles. So it's something to consider. In terms of learning, Judith, yes, you're always encouraged to learn this. You know, please. It is hard work, but it can work.
You talk about 3 weeks. What time of year was that? What was the temperature?
Good question. It was about a month ago.
Yeah, so it's getting cooler now, so it's going to be slower.
Yeah, we're still going to be trying to grow this out over winter. You know, fungi especially, they don't mind colder temperatures. We had fungi growing in the fridge, so.
Mm-hmm.
Another option would be to use your green waste compost, if you can access it, and use it as a base, as a bedding for worm composting. That also gives you a fast turnaround.
Sorry, was it a question or a statement?
Statement.
Statement. there you go. Ian, sorry, you also had a question at the same time?
Yeah, just are there some good resources on how to go about making compost, and what is a good system? Because obviously there, I mean, I've read some of Nicole there's
Masters.
been in love with Nicole Masters' book and read some of her stuff about compost making, and there's clearly, you know, there's lots and lots of different types of compost and lots, you know, you've got to know that. But are there some good resources on how to do it on a farm? I mean, should it be done on concrete? Should it be done on soil? Should it be done on straws? You know, what are the fundamentals?
So first of all, you've asked a question, and there's a guy popping into view from time to time. His name is not Gianluca. His name is Adam, and if he unmutes, he'll be able to answer some of these questions. That's the person you want to speak to. There we go.
Hello. Yeah, you've got lots of different options. I guess it depends what materials are available to you and what your holistic context is, so to speak. Obviously, we're limited by legislation with large piles, right, Daniel? You're meant to do it on concrete, but it would be more ideal to do it in contact with the earth, as far as I'm concerned, you know, try and draw some of those native microbes up from underneath the pile. I'm not sure what your limitations are as a farmer. Do you know, Daniel, like legislation-wise?
So in this legislation, I think has more to do with waste, but it would be a conversation with your local environmental agency people to double check, you know, how much material you can actually store at the same time. Organic matter, you know, on the farm, but people have piles of manure, you know, and things like that, you know, that they've taken out from the cattle sheds, and that can be sitting for months oftentimes. So it seems that if the material has been produced on the farm, you've got much less limitations than if it's material that's been brought to the farm. If you're bringing material to the farm, it's classed as waste. And the moment material is brought to the farm and classed as waste, then you're dealing with legislations, and then you've got environmental agency on your tail. But if you're producing the input materials on the farm, you're solving two problems. First of all, you're not really have to be dealing with that legislation. The second thing is you control your inputs.
And you have lots of different options. You have static composting, you know, active thermophilic composting, trying to cycle all the material through a hot center of the pile, whether it's a windrow or some kind of stack. Then we have a process we've been working on here, which is a 16 to 22 day process. We've been conducting that on concrete, and it makes some pretty good numbers as far as nematodes, flagellates go, things that are going to cycle nutrients for you in even in possibly degraded soils. So there's a good sign from our initial trials with it that it works in the ag soils, even though, you know, they're 200, 300 psi, not very far down. So yeah, maybe we could organize a workshop for those who want to learn. We could do something like that, and we could introduce you to our more faster systems, and then maybe the static composting, which takes longer, but as long as you set it up right, is pretty much guaranteed to get you great protozoa and possibly, you know, within a year, decent fungi. It really depends on where you are and what you use, and again, the whole holistic context of the thing. But yeah, we'd love to teach people and talk about it all day long. We love it. So
because there are, I mean, even just on this small congregation on this call, you've got Tom from Dyson, you've got Mark from Velcourt, and yeah, we work with lots of different types of farms. You know, I'm sure that, you know, there's an appetite for this kind of stuff, and it is the lack of knowledge, and not just the lack of knowledge, but the lack of advisors to give the knowledge is part of the problem why we're not doing more of this stuff.
Right, and everyone's going to be so different, so it's going to have to be tailored for the individual as much as possible. So yeah, I mean, Daniel could maybe organize that. We could do a separate drop-in session or some kind of workshop, whether digital or in real life sometime. We'll have to think about that.
We will have to think about it. I mean, the thing is that there's a lot of stuff digital. You know, the ultimate is to go to Dr. Ingham and go through her course. But what we basically found was that is doing things is unfortunately necessary to learn this stuff properly. And I think you know, some practical workshops may be possible to do. Maybe even we should consider that over winter, because this is when people have time. Is that a good idea?
The other thing would be to identify the nearest green waste facility to you, go there, check it out, see if it's good enough for you, see how much plastic it contains, and then maybe we could supply the inoculant to bring that green waste to life. Since it's already been put through the process to remove pathogens, all you really need is the good microbes, which will come back in like extended amounts of time, but with our inoculum, it will come back a lot faster and it will get to work. So that's another way.
Another option to get going this winter, given that you've got maybe 6 months or so before application, is just to get some wood chip on site. If you can get hold of some willow wood chip, for example, from some local short rotation coppice, then all the better, basically, and just windrow that like now, basically, in the field or some spot, basically. If you can cover it with some compost fleece, great, but you know, that's sort of optional. And the longer that sort of stands, basically, the better the sort of base you've got for sort of upcycling, sort of come next year, for example.
Personally, I think as a farmer, if you want to make it more wide scale, is getting the benefits of the compost from the extracts and the teas, actually trying to physically apply 2 ton a hectare of compost and going to hauling compost from waste sites, etc. You know, to get to the larger scale, I think it's putting it down with a drill or put it on with a sprayer. That's where you're really going to make a big difference.
I completely agree with you, Andy. And we're actually speaking with a very large green waste composter. I want them to run a project where we build out large-scale growing out of our biology in their compost. We skim the biology, so we literally just take the extract, and we provide that to farmers in a concentrated form. So we have actually, Adam, have you got any of the goop with you over there?
Well, there's some diluted goop in front of me. Yeah, it just goes into solution. Well, it just did it, actually. I didn't. It was just all at the bottom, and now it's in solution. Like it works really well. It's not going to clog any sprayers or anything. It's super
Actually,
easy to use. It's
hold on
delight to use. In fact, we can do it right in front of you right now. We got a little bit.
I'll show you something. This is what I'm going to do. Give me a second. I'll get a fresh spoon and stuff. I've got it. I'm doing it.
Close the door.
One second. Nice, good clamp. Can you close the fridge for me?
Yeah.
Thank you, sir. Can you close the door behind me, please?
It's the big moment, look.
This is
It's the humble goop. I'd eat it.
Give them a good look at it.
I wouldn't drink glyphosate, but I'd eat that.
This is the goop. This is purely microbes extracted with a little bit of organic matter that went through the sieving process. But basically, this goes into water like this.
You don't even need that many stirs.
So we're basically down to, in terms of application, I think we're down to something that should easily go through anything, and it's just something that we could provide as a product that's actually very easy to apply.
It looks magical on the microscope. Magical.
We should do some. Can we look at it under the scope now?
Ooh, is there a way we could do that?
Yeah, he's got one next to him. We've got one next to us.
Shall I give you a good dilution, Daniel?
If you connect your camera, you've got all the gear over there. Can you do that?
Yeah, you want 10 to 1.
My hands are covered in the molasses, so
Get that smooth.
Just there you go.
I've been on quite a few webinars recently, but this is definitely the most exciting. It's got the most exciting the figures. We start mixing up goop and start sticking it under a microscope. This is what we need. This will revolutionise the take-up of this because it's much more interesting. Just talking about it.
It's very exciting, this product. It's a lot of work has gone into it, and yeah, we looked at it yesterday for a good long time, and it's very nice to see. We've got big fungal feeders, huge amounts of fungi, and like I say, it goes into solution rapidly and it won't block the sprayers. So now we just have to produce it on a massive scale. Which is a different ball game, but good fun.
And this is a product of frustration, by the way, because last week when we were preparing for the drilling, the guys were really trying hard to get the extraction process to go down, and nothing was working. And I was busy with some calls. I think I was speaking to some investor that I shouldn't be speaking to anyway. And the guys were getting really frustrated with it. And it took us until I think 10 p.m. me and Tom, just to make sure that that the pumps were working and everything was working. And then in the end we came up with this product that we're jumping with joy. And we finished at 2 o'clock in the morning, basically, seeing that we can't really coat the seeds. But we've got in the end, we've got 40 liters of this goop, and we're just getting ready for a spray on Friday because that's the only window of weather we've got to apply for our trial. But yeah, we've got stuff to show now thanks to it.
Would you expect the microbes to survive if applied to a pile of digestate in the field, solid digestate, or manures or dried poultry litter?
Good question. If it's a pile of material, organic material, yeah, it should do. There would need to be some toxins to start killing it. But if it's organic matter that's got moisture in it, I don't see a reason why not. Then it would be a case of, which microbes are selecting for the foods that, basically are in that pile. So yeah, good question. We would need to, try that.
Thank you.
Everybody's now waiting, and all of the microbes are going to be in every other pile of part of the goop, basically, because everybody's waiting for Tom to do that. That particular drop will have nothing in it.
Can I ask a question, please?
Yes.
You're talking about the trials, and there's three bits to it, like the biology, the conventional farming, and the control. So is the control with no chemicals, no chemical fertilizer at all?
Yes, exactly. This is why I'm looking to get money from the outside to limit the
so
loss.
That's going to be a fairly big loss to the farm, that section.
Yeah. So we're still working on that. We're hoping to get some additional sponsors, but at least I'm hoping to make sure that, because we're reducing, if we can get the grant, we can reduce the total cost of the trial down to 30% to the farmer. And so hopefully that's going to be one thing that if you think in the larger scale of things, if we reduce your cost of inputs by 70% on that trial area, hopefully that will even out the control. So we need to work out the numbers.
Yeah, the prices you gave me when we talked about it, the cost of the inputs doesn't frighten me. What does frighten me is doing the proper monitoring, scientific monitoring and analysis of the samples that need to be done to prove that it does or doesn't work. That's the expensive bit.
That's going to go down as well, though, because the grant that we're trying to get basically is the one that we're basically. I'm basically trying to find as many sponsors of these grants as I can.
Yeah.
You know, any ideas for organizations that we can contact that could sponsor your trial, that that are local to you, that you know of, any people that you know of, any people on this call that that have got some good contacts that will be willing to put up some money to the kitty for to help with some of these costs. That is obviously very, very welcome. So,
to me, I think I would rather have as much as possible of the analysis covered. I'm not worried about the cost of the inputs, the biological inputs or buying the compost you're talking about. I'm not worried about that. That's not, to me, that expensive per hectare or per acre, however you want to look at it.
mm-hmm.
What is expensive is the monitoring and the scientific analysis. That, to me, is.
And if I can just say for everybody's benefit, the reason Judith is not worried about the inputs is because we already talked about the way of reducing the cost of inputs by trying to grow out the biology in the green waste compost. So we basically try to we will also basically include a version into this whole offer of what we propose to Judith. And this is, in essence, a double trial, because we basically have to test two things. One is the efficacy of the biology on the ground, but also will this biology be able to propagate through a green waste compost successfully? On 10 hours, 12 hours drive away from us. So that's the sort of thing. That's the sort of thing that adds to complexity, actually, in Judith's case. But it reduces the cost of inputs quite drastically. There's a fellow.
He didn't have to go very far, did he?
No.
Can I just input there that if there's a research institute within the collaboration, they will do all the analyses.
Well, we have to do.
You do yours, and then we would do ours
as
from
well.
the field. So we would share that analysis and the cost of it.
So in terms of Judith, for instance, in terms of what Sheffield University will be doing for us, there's a lot of DNA work that we would like to do, and that is completely free to the farmer, because they will be completely covered by their grants and the grant that we're trying to get. If we get the grant, this is the part of the thing that we don't know yet. But if we get the consortium, hopefully we will. The part that doesn't get covered 100% is our work, and so this is where the 30% needs to come from somewhere. He's looking at a nematode, and we still haven't. I don't see the mouth yet. It's not the. I think you're muted right now, Tom. If you guys unmute Tom, then we'll be able to hear him. Can I unmute you?
Yes, you can do that.
Tom,
You can
unmute.
request it. If you're the host, you can request him to unmute.
Oh, there we go. He's unmuted.
There we go. Yeah, pretty confident he's a fungal feeder. He's got this kind of well-fed look to him, much fatter than fungal feeders that we usually see, and much longer. So it seems as though this guy's almost a full 2 fields of view.
Oh, there we go. There's the stillet. You can see that now. There's the stillet.
So how
many times is that magnified?
That's 400 times. Oh, sorry.
There we go. So this is the sort of fungal feeders that we found in this material that we've produced. The interesting bit to when we look at samples, basically the technique that we look in samples, we'll take a sample of soil or compost, We'll dilute it 5 times to be able to have a water suspension, and then we look at the drop under the microscope. We have never seen an abundance of these fungal feeders when we were doing that, even if we were looking at the same material as this. So what we basically are thinking of doing, the fact that you've got this material so concentrated allows us to then make a decision, you know, how concentrated do we want to put this out onto a field? That's one thing. But the second thing that makes us think about concentrating material is actually also concentrating soil samples, because we have a massive issue with soil samples in the sense that we have soil samples being sent to us and they all come out bacterial with this methodology. And we know that this methodology is not lying. We understand, you know, why they're bacterial. And we also have seen that when we were applying our biology, there was a response large enough from at least the nematode population that we then had a feedback, you know, when we were testing afterwards under the microscope. But what we're hoping to do is to be able to give you a better picture. And it's not something that we've developed yet, but it's something that's for the future we'd like to develop, where we actually do a concentration of what biology could be in your soil and actually have a better picture qualitatively, not just quantitatively. So that's on a side note, basically. So, but yeah, you've got a, I think this is 100 times magnification right now.
This is 100 times. We're just doing a scan, see if there are any others on this drop. But you can see loads of little strands of fungi all over.
We can't exactly make them out though, because if you're looking through the oculars,
yeah, sure,
the camera is not picking up the same.
Looks better already.
But just the presence of these fungal feeders is indicative of an environment that's full of fungi, right? So
yeah, and especially the size of them. They're full 2 fields of view long, if not longer. I think Adam yesterday saw one that was nearly 3 fields of view long. So that really shows they're in an environment with plenty of fungal food. Otherwise, they wouldn't have gotten that size.
But Daniel,
sorry,
can I just ask a similar question to this? The video I sent you last night of our extract, the fact that I only looked at 1 drop and 1 sample from my thing, is that possible that there just wasn't any nematodes in that drop, or just what? Because the nematodes was in our compost, but we couldn't see any in the extract.
Well,
you got any ideas why?
Yeah. I mean, we would need to look at the whole chain, basically. You know, from the compost to the extract. We would need to look at everything. And then basically ask ourselves, you know, what is happening? Are they being diluted and you're just not seeing it, or are they being lost somewhere? And so you sent that video, and that was really showing nothing but bacteria. And so yeah, so this is something to look into as a whole chain. And also, how far did you dilute it though, as well? What was sort of the application rate that you've put into that?
Fairly dilute. Because it was I doubled what David Johnson suggests for his compost, because it was going on the surface rather than down with a drill. So it was 4 kilograms per 200 liters.
No, that's tiny. I can tell you now that you've diluted too far. We use 70 liters of compost per hectare these days. And so, you know, we would have the water in our tanks going to be just, you know, completely dark. So it's the dilution factor. The dilution. Dr. David Johnson, by the way, uses anywhere between 200 and 400 pounds a hectare.
It's not what he says on his all his videos. He says two £2 per acre for his extract.
No, 2 to 400.
Well,
Daniel, have you worked with lower application rates?
The first one, the first application on barley, I think, was probably the very poorest that we had, and so that was just brown water that would probably just have some nutrients in it and not really much biology. So that's why we've upped it. But the reason I'm saying that the Dr. Johnson says that because we hear that from Dr. Ingham directly, and they work together. So this is something that yeah, I wouldn't go with £2 an acre. It's too low.
Yeah. So what rates have you actually worked with specifically?
Well, we just went straight up to 50 kilograms a hectare.
Okay. So you've not worked with less than 50 in the field?
No, unless you're making compost tea, then you can start with a lower amount, but you're not producing everything. You're just producing bacteria and flagellates usually.
When you say producing, what does that mean in this instance? If we're just talking about extracts?
Yeah, if you're working with compost teas, you can start with a smaller amount and just produce bacteria and flagellates. But in terms of extracts, you're just diluting the biology too far. And the thing is that they have to make their way to the root zone to be able to be fed. And so if you're diluting them that far and then you're spraying them with an overhead sprayer, fair enough if you've got an injector. But if you're putting an overhead spray, they have a very small chance of actually getting onto the root zone. So this is why we basically want to get to the point where they're coated on the seed, they're plowed into the soil, the first time round, if the soil has been plowed, injected into the soil, just applied to the root zone as efficiently as we can, so that we can use lower application rates. But I think we still need to put out quite a lot in the first days anyway, so that they have a better chance, of partially surviving. That's another set of questions that we need answered next year and the next 2 years maybe. What survives in the soil after application? Because we think the attrition rate is actually quite high. I think Tom is still looking for some more pals of that one fungal feeder. He's going to be lucky if he finds it. But all these fungal threads, there's a lot of organic matter here, but there's lots of fungal threads here. The one thing we don't get in this group is Protozoa, and that's something that we're still working on. We've got some ideas of how to include that, and also this group doesn't include the bacterial nematodes that we produce in another process that can be added to it very easily as well. You're muted.
We've got some other processes that we're experimenting with, growing out more fungal feeders in this goop mix when we dry it even more and apply sort of environmental stress, such as cool temperatures, and that drying process combined possibly would kick them into feeding more and bulking up on the food, the fungal food that's around them. And essentially you end up with bigger fungal feeding nematodes as a result. It's still experimenting with, but we had an accident the other day where it seemed as though that was what was happening, and we're quite excited about that one as well. So it's constant developments, really.
Niels, thank you for that. David Johnson's directions. That's a long piece of fungi there. the one thing 4 years ago when I was starting learning this stuff, you know, I would get told about the fungal component, and then I would buy my microscope and start looking, and I wouldn't find any. And so nowadays, basically, it's not a day by when I can actually have a look at the fungal strands, and lots of them. So, oh, there's another fellow, some younger one.
Can I ask your fungal feeders fungi specific?
That's a very good question. Adam, you were reading something about predatory nematodes.
Yeah, we don't know is the answer to that question.
Mm-hmm.
We have a lot of fungi in the product, so they seem really happy.
Mm.
They seem really, really happy, which is great.
Yeah.
And
not many people know about what fungi is in soil.
And also actually, Helen said that they're finally looking at our bacterial feeding nematodes from the DNA perspective, right?
Yeah.
So that was, you know, we sent that sample 2 months ago or something like that. Since then, you know, we found that in the same process we were able to grow out different bacterial feeding nematodes. So we're kind of hoping that we're going to get to the point where we'll be able to grow out, you know, different species of bacterial feeders, different species of fungal feeders, different species of protozoa, using same process, but maybe different foods. We don't know yet.
Mm-hmm.
Again, part of this this whole drive, you know, having Sheffield on board, if we can get all the money from the grant, then basically Sheffield will be tasked to actually tell us what we're actually growing.
Mm-hmm.
And the question about, you know, what do these fungal feeders feed on is very valid. I don't know if I said it in the last one, but the way I like to look at this is that, you know, we're a big complex organism and we can eat a variety of foods, but these microbes, they're so tiny that they can only metabolize very specific foods. And so that's why it's very specific, and that's why when you hear about, for instance, entomopathogenic nematodes, they can be very specific in terms of, you know, what hosts they populate. You know, if you're speaking about bacterial feeding nematodes, they've got specific bacteria they will eat. When you're talking about predatory nematodes, they've got specific root feeding nematodes that they will feed on. So they will not be able to even, you know, control the whole population of root feeders. They'll be only really controlling a specific species. Mm. And so this is why Dr. Ingham is pressing so hard on diversity, because then we should be able to cover all cases.
Yeah.
Me being anti-dogmatic, I still hope that, you know, we can at least have a knowledge of what we're producing and what is happening in the field, so that we know that we're covering all cases, or we're actually missing some cases.
Yeah.
And so that's the sort of work, you know, that still needs to happen.
Yeah.
You know, from the scientific perspective,
yeah.
from the marriage between scientific and the practical, to be really able to answer those questions. I want to stop this whole hopefulness about soil microbes that everybody's talking about, you know, that it's just going to you know, just make the compost and apply it and you'll be fine. I think we need to know more. So.
Yeah. Mm-hmm.
And I really don't know how Dr. Johnson is getting some of the results of fungal to bacterial biomass ratio. I don't know what he was actually using to measure. When you know, his videos on his YouTube channel just show bacteria, nothing else. You know, the application rates are so low.
Isn't it all about spore counts?
I don't know. His videos don't even show spores.
Which videos are you talking about?
On his channel, he's got some old videos basically on his channels of this stuff, and it doesn't even show spores.
Of
He's got microscopy videos. He's got microscopy videos there. So I don't know, it's like the videos basically show some bacteria. And
so what about the DNA analysis that he publishes?
Well, that's the interesting bit, right? That's the interesting bit. But the DNA analysis also looks only on the bacterial groups. The DNA analysis he's got doesn't cover protozoa, doesn't cover nematodes, doesn't cover fungi. So it's so what we're interested in is we're interested in the whole soil food web and actually getting that. So yeah, I'm very eager to replicate his results. I'm very eager to be storing 10 tons of carbon per hectare per year. With this stuff. I just want to know more. I want to answer all of these questions that are just burning inside my brain. So
there's no data on spore taxonomy then in his.
No, I've not seen anything published. I think
no,
I have to just reach out. I might have to just reach out to him. Because the thing is that the chart, for instance, I sent to you. This is something that that I know that he's made available. Is a very generic chart. It doesn't tell you these guys are beneficial, and some of them might not be beneficial. The spore chart that he sends. If you look at the spore chart, for the rest of the people on the call, have we stopped presenting now, Tom? Oh, there's a new drop. There's a new drop coming. So stand by. So the spore chart that I'm talking about, I might be able to pull this up when they guys finish and show you. It's a spore chart of all types, of almost all types of like generic spores. But when you look at like the bottom of that chart, the smaller spores are actually the basidiomycetes, which in general would be more beneficial because that's the fruiting body guys. So it's but the rest of the chart is could not be beneficial to plants, and so we could count these spores, but it doesn't tell us if we're actually going to get a good result. So it's still an open question, and the work with. Speaking of spores, my other pet pain is arbuscular mycorrhizal fungi, which currently mostly are produced in cut pieces of roots. But I really want to find a product that sells spores. They are much more difficult to produce, because then we definitely control the AMF fungi. Then we basically put that onto the seeds. Job done. I'm happy. We put the known known bugs in a jug on the seeds, and we're happy. We're applying the soil food web, the rest of it, and we're happy. So that's the sort of thing. That's the sort of thing that I think, in terms of the general picture, we'd like to see. But yeah, another fungal feeder. This is, you can't imagine how exciting it is to me to have a material that the guys have no problem with finding fungal feeders. Like, you guys have been sending us samples of compost, and we would be jumping with joy when a sample would come in with fungal feeders in it.
Even just a nematode.
You're still muted.
Even just a nematode, let alone fungal feeders. These are highly developed and should really only be in systems of high fungi. So fungi is quite a hard group for us to get, and there's always been sort of the slower group, with the secret element sort of being time. And we're experimenting with all sorts of ways to kind of boost its growth in our piles. But to see fungal feeders like this indicates a high fungal environment. Otherwise, they wouldn't be there. I know I'm repeating myself a little bit, but it does make us really excited.
Anyways, I'm going to actually just say, is there any more questions? Because we've run out of time. Again, sure. I don't mind, but I'm mindful of everybody's time. It's late in the day. People want dinner as well. So is there any more questions that we could answer? If there is no questions, then I will encourage you to think about connections for creating a group of farmers. think about potential people that you might know of who could put some money to the kitty as well, so it could be made easier to run these trials for everybody. I've put into the chat as well, if you need to send us samples, we encourage you to send us samples of your compost, and also send us samples of compost that you think locally might be useful to you. And so you can get that tested, so you can have a baseline, and consider the trial even before this trial, where you buy a bulk bag of compost. This is what we proposed to Judith, to buy a bulk bag of compost. And in her local area, a bulk bag of compost, 850 liters of compost, green waste compost, cost 40 quid with VAT. And then buy 200 milliliters of our concentrate and put it in there. And that we would sell that for 20 quid plus VAT. So that's something that then you would need to send a sample at least once after 3 weeks. I think we would really want to see it 3 times, after a week, 2 weeks, and 3 weeks, because then we would be able to tell if it's actually developing. And that way, there's a possibility that you might have an inexpensive way of growing out biology very simply. And so we propose that you consider testing that hypothesis. And what we will do from our end is basically work on making sure that we've got this product where it's got the fungi, the nematodes, and the protozoa. The protozoa seem to be the most difficult to put into that simple product, because they're just too small and they just get sieved out with bacteria. However, the important bits are the nematodes and the fungi, and the protozoa are usually present in your compost. So when you make compost, you actually grow out the protozoa quite successfully, and we've seen that in that leaderboard thing. So continue making that, and yeah, send us more questions. And if that's good, we're going to finish here. Thanks everybody for the participation today. And if you're available next week, we're running this until the middle of December, basically. So every week, 4:30 Wednesday, and this recording is going to go out in probably around Saturday. Thank you very much.